Hello and welcome to a quick video on actin filaments, one of the filaments that make up the cytoskeleton of the cell.
The actin filament consists of a two-stranded helical polymer of the protein monomer actin. The filament is 5 to 9 nanometers in diameter and one full turn of the helix takes up 37 nanometers. The actin filaments have a central point, a focus in the cell from which the filaments radiate. The filaments form as long fibers with 70-degree branches to create a web-like structure. The branching process is controlled by a complex of proteins called the actin-related proteins. These proteins allow for the elongation of actin filaments in one direction and can also attach to the side of another actin filament to form the web-like structure. The actin filament grows by the addition of actin monomers to the positive end of the filament, that is, the end of the filament that is not attached to the actin-related proteins. Actin filaments can determine the shape of the cell and are attached to the plasma membrane. Actin can form stable structures, such as in muscle, where they are involved in contraction, or can form temporary structures that are involved in cell movement. Actin filaments can be viewed as forming four types of structure in the cell. 1. Contractile bundles, as seen in muscles. This can be viewed as loose packing of the filaments with the protein myosin-II between the filaments. And it is myosin-II that provides the contraction. 2. Gel-like networks, for example, filamin. Again, these are loosely packed. 3. Dendritic networks of branched actin molecules. And 4. Tight parallel bundles such as in fimbrin. For controlling cell shape, the rigid actin structure is attached to the plasma membrane lipids by myosin-I and calmodulin proteins. Now, this nicely brings us to the myosins. The myosins, of which there are 8 different types, are motor proteins that use the energy from ATP to walk along the actin filaments. The myosin filaments are involved in muscle contraction and the movement of molecules and vesicles around the cell. In terms of breaking down actin, this is controlled by the rate of subunit dissociation. The subunits will independently dissociate from the growing chain, but if new monomers are added faster than the old ones dissociate, then the chain will continue to grow. That is, chain growth is dependent upon the concentration of free monomers. The actin filaments can also be broken down by the severing protein such as gelselin, which can break the long filaments into smaller filaments.
Okay, so in this video we've looked at actin, which is one of the filaments that makes up the cytoskeleton. We've looked at the function of the filament, how it can be assembled and broken down, and how it can be organized in the cell.